Everyone, and welcome to Lab 4, Cardiovascular Anatomy and Physiology. Now, when I took Bio 162 many years ago, this was one of my favorite labs because the heart is such an incredible organ that does incredible things. So I'm really excited to teach you guys about the cardiovascular system along with the respiratory system because they kind of work hand in hand. So yes, as mentioned, cardiovascular is going to include your heart, the veins and arteries, and then the respiratory system is going to include your lungs and the tissues involved with that. So activity one, you will be familiarizing yourself with different structures of the lungs. The bronchi, which are the trachea moves down into these branches here. These major branches are your bronchi, or singular. This is one bronchus. Then your bronchioles. Those are going to be these smaller branches that break off of the bronchi here. So here's a bronchiole, here's a bronchiole. And then your alveoli are going to be at the tips of the bronchioles. So if we zoom in on that here, these alveoli are these small little grape-like structures. And so a single of these structures is an alveolus, plural is alveoli. And so inside each of these is where gas exchange occurs. And then diaphragm, singular diaphragm, is this structure here. It's a muscle that facilitates our breathing, and it's really important to have that intact. In fact, not to give it away, but I did post a case study video on your lecture PolyLearn of a Rottweiler who came into my vet clinic that I work at who was having some breathing problems. And I'm not going to give anything away, but it's open for you guys to watch. It's not required. If you do have a problem seeing a dog undergo surgery, then maybe don't watch it. Nothing to worry about there if you don't want to watch it. Like I said, it's not required. It's kind of just for funsies. Okay, so as mentioned, when we breathe, air enters our trachea into our bronchi, and then moving into these smaller stems, the bronchioles, which then go to these grape-like structures on the end, the alveoli. That is where gas exchange occurs. So this is a single alveolus, and wrapped around these alveoli are capillaries. These capillaries are bringing in deoxygenated blood from the heart, and this is where gas exchange occurs, where CO2 leaves and O2 enters the bloodstream. And so then oxygen-rich blood returns to the heart. And we'll talk about this more once we get into the heart dissection. The diaphragm facilitates our breathing. So when we inhale, this diaphragm is actually contracting downward. When that diaphragm contracts downward, this negative pressure is in the thoracic cavity. And so when you have a lesser pressure than atmospheric pressure outside, air is going to move in and fill up our lungs. On the contrary, when we exhale, the diaphragm relaxes and creates this positive pressure on our lungs that pushes air back out. And so just to show another figure of that, when our diaphragm is contracting, when we inhale, we are creating that negative pressure in our thoracic cavity that allows air to move in and fill up our lungs. And then on expiration, when that diaphragm relaxes, it creates this positive pressure that pushes air back out. Don't think about it too much. When I learned about that as an undergrad, it kind of wigged me out a little bit to just think about my breathing. And then activity two, we're going to be diving into some histologies, so microscope images. Now, we've already supplied those images to you on the lecture PolyLearn. So you are going to be responsible to compare and contrast a healthy versus diseased lung, healthy versus diseased blood vessel, a healthy versus diseased erythrocyte, aka red blood cells, human versus reptilian blood, and then you're going to be looking at a cross-section of an artery, vein, and a nerve, and you should be able to tell the difference between the three and why they look that way. So as mentioned, this can be found on your lecture PolyLearn under Lab 4. If you go down to slide images, you will see all of these slides here. Now, before I send you on your merry way to go look at those slides, let's talk about them a little more in detail because some of them can be a little confusing, especially the lung tissue one. So let's talk about that one first. So right here at the beginning of the animation, that is healthy alveoli. So right there, you have your healthy circular alveoli, but over time, notice how they seem to almost melt into each other. Those membrane walls begin to break down. Sometimes they rupture. And this is the case in emphysema patients. So over time with emphysema patients, that membrane in the alveoli breaks down. There's less surface area for gas exchange to occur and a buildup of air in these alveoli can occur. And so oftentimes emphysema patients complain about not being able to breathe or not being able to get sufficient oxygen. It's because these alveoli are damaged. So what that might look like on a microscope slide is this. And so this is what you guys are going to be looking at. What you see here on this slide is actually a combination of healthy versus unhealthy lung tissue. And I went ahead and put a mark down the middle that separates the unhealthy versus the healthy. Can you guys guess which side might be unhealthy versus healthy? Well, remember that I mentioned that as these walls of these alveoli break down, there's less surface area. So notice how on this side, there's less pink. There's less tissue for gas exchange to occur. Compared to this one, there's way more tissue over here that facilitates that gas exchange. And so that's the big difference that you guys need to know of, is that this is diseased lung tissue because there's more air, there's less tissue for gas exchange, versus the healthy lung tissue over here, there's more tissue. And while there are air pockets there, that's totally fine, it's the lungs, but there's more tissue for gas exchange to occur. I think it looks like swirls of cotton candy personally, but you guys should be able to know the difference between diseased and healthy. Okay, now let's talk about the diseased and healthy blood vessels. Specifically, we're talking about the arteries. So on the left over here, this is going to be a normal artery or a healthy artery. Notice that blood is able to freely move through this vessel. Now, on the contrary, when you have a diseased artery or atherosclerosis, it's a mouthful, there is a buildup of plaque that occurs in the vessel, thus inhibiting or slowing down the movement of red blood cells. And so they can actually build up here, and this can lead to more serious problems. And so things that cause atherosclerosis could be things like one too many Big Macs, or interestingly enough, chronic stress over time can actually lead to a buildup of plaque in your arteries. And so I want to warn everybody here that when you go onto your lecture PolyLearn, you're going to see a cross-section that looks like this of atherosclerosis, but we do not have a cross-section to compare it to that is healthy like this. So to help you guys out, I've supplied in this next figure a healthy versus a diseased artery so that you guys can compare and contrast. So notice how the opening of this artery here, this is called the lumen of the artery. Notice how it's nice and big and open. This allows blood vessels or red blood cells to move through freely. But in this artery, you see how it's building up right here? This is all that gross, icky plaque that is building up in the artery. And so over time, this will build up more and more and more and inhibit red blood cells from moving through freely. Okay, so next, we are going to be looking at healthy versus disease red blood cells. So over here on the right, this is healthy, a healthy sample of red blood cells. A lot of students get concerned about this. This is totally fine. We should have these in our blood. These are white blood cells. We will talk more about these in detail in our immunology lab. This, on the other hand, is not healthy blood. This is from a patient who has leukemia or cancer of the bone marrow and the blood. And so just notice how these red blood cells just do not look happy. They're undifferentiated. They kind of look sticky and clumpy. That's a result of the cancer. So be able to differentiate between the two of these. Now that we know what healthy blood looks like in a human, we're going to compare that to reptilian blood. And so you guys should be able to identify which one here is reptilian blood. But not only just that, what are these purple things in the middle? And why might one sample have them and the other one doesn't? And so that is for you guys to investigate and to figure out. And if you're having trouble with that, go ahead and come to office hours and ask us about that, and we can chat. Okay, and then lastly, you are going to be looking at a cross section of an artery, vein, and nerve. All three of those are in this cross section right here. And you should be able to differentiate between the three. So I'll give you guys a hint. Okay, leading into our discussion on the heart, which is coming up next, arteries are really thick-walled because they need to withstand so much pressure of the heart being forced onto them because blood is being carried through the rest of the body. So being that they're withstanding so much pressure, they're going to be really thick-walled. Now, veins, on the other hand, they're not thick-walled because they're not enduring that kind of pressure that the arteries are. So they're thin-walled, and when a cross-section is done on veins, they oftentimes collapse and they look smushed. And then nerves, they don't have a pretty little lumen like arteries do. They kind of look like salami slices, in my opinion. So I will let you guys know that there's probably more than one nerve in this cross section here. So be able to identify those three and distinguish between them. Okay, let's talk about the heart. This is fun. Okay, so activity 3 is our heart dissection. Now, unfortunately, we're not able to do that in person, and I'm hoping nobody has any readily available hearts to dissect. But let's talk about that. So the goal of this is to understand the flow of blood as it enters your heart, goes through pulmonary circulation, back into the heart, and then goes through systemic circulation. You should be able to follow blood step by step as it enters and exits the heart. And you can find these step-by-steps on your lecture PolyLearn. There is a PDF posted, although I'll tell you now that it might be a little confusing because that step-by-step is usually if you were dissecting the heart yourself. But I'm going to be posting 2 YouTube videos, one of myself talking about the structures that you guys need to know in detail, and then the other one is going to be somebody who is actively doing the heart dissection as blood enters the heart and leaves the heart. So both of those videos are really important for you guys to understand the concept of activity 3. So let's talk about this just a little bit before we move on to activity 4, and then after that you can watch my video on the heart dissection. So I mentioned something about pulmonary and systemic circulation, or the pulmonary and systemic circuit. What does that mean? Okay, so to go over this very broadly, blood is going to enter the heart via a vein. Right? So veins are going to be carrying deoxygenated blood back to the heart, where arteries are going to be taking blood away from the heart and delivering it to the whole body. So deoxygenated blood is going to be entering via the superior vena cava as well as the inferior vena cava, but we're not holding you guys responsible to know the inferior. You guys just need to know the superior vena cava. So deoxygenated blood is going to enter into the right atrium, right ventricle, and out through that pulmonary artery. From the pulmonary artery, it's going to go to the lungs where it gets oxygenated. Now, what I just described to you right now is pulmonary circulation. This is your pulmonary circuit where deoxygenated blood enters the heart and leaves again to go to the lungs to get oxygen. That's it, pulmonary circulation. Okay, so now blood is in the lungs and it's getting oxygenated. So remember that we talked about this a little earlier where oxygen is entering each of these alveolus or alveoli, and wrapped around a single alveolus are these capillaries. And deoxygenated blood from that pulmonary artery is entering the lungs and gas exchange occurs. And so when blood leaves the lungs, it's oxygenated and returns back to the heart. And so blood is going to enter via the pulmonary vein into the left atrium, then into the left ventricle, out through that aorta, and this is where blood then enters the rest of the body to deliver oxygen. This right here is called systemic circulation, meaning that blood is going systemic throughout the entire body. So that overall is just a broad walkthrough of how blood goes through pulmonary circulation and systemic circulation. This is another figure that sort of just shows that in a generalized overview. Pulmonary circulation goes to the lungs, gets oxygenated, returns to the heart, and then the rest of the body. Okay, so next up, we're talking about arteries and we're talking about veins, but what is an artery? What's a vein? A lot of people think, well, an artery has oxygen-rich blood and a vein has deoxygenated rich blood. Not always. There is an exception to the rule. So if we look here, this is an artery and this is a vein, and it's red because we think arteries have oxygen-rich blood, and this is blue because we think veins have deoxygenated rich blood. Well, if we go back to this figure here, the pulmonary artery is carrying blood away from the heart, but it's deoxygenated blood. And the pulmonary vein is carrying blood to the heart, but it's oxygen-rich blood. And so these two vessels here are the only two vessels in our body that are doing the opposite of what we would think they'd be doing. While arteries, this artery is still carrying blood away from the heart, it's deoxygenated blood. And the pulmonary vein, while it is bringing blood to the heart, it's carrying oxygenated blood. So don't get confused about that in the future. These two are the only two vessels that they're swapped. Okay, so going back to this figure here, how we like to think of arteries and veins is that arteries are really thick-walled. Remember that they have to withstand that pressure, versus veins are really thin-walled. And not only just that, but veins are going to be carrying blood back to the heart, and arteries are going to be carrying blood away from the heart. So that's the difference between an artery and a vein. So let's talk more about the heart. If this is the front view of the heart, the anterior view, so front is anterior, which side is more muscular than the other, the right or the left? And so in order to tell the difference, this is a model of the right and left side of the heart. Here is that right ventricle. Notice how thin-walled that ventricle is versus this left ventricle is really thick-walled. And so that's the difference right there. The left side of the heart is going to be way thicker than this right ventricle. And that is because the left side of the heart is pumping blood out to the rest of the body. So it needs to be more muscular to really pump out blood, versus that right ventricle doesn't need to have a thick wall because it's only pumping blood to the lungs. Now I know this is really pretty. This is a gorgeous model of the heart, but this is not how it always looks. This is how you guys should be able to identify which side of the heart is more thicker than the other. So look at that wall. Notice how this wall right here is really thick. Let me get my pen. Really thick. This is a thick wall. So this is the left side versus this side is a lot thinner, and this is the right side. So here is that right ventricle, and then here is that left ventricle. Notice how thick that is, and then here's your aorta. Okay, now we're not quite there yet, so don't fret. You will need to be able to identify that on a picture like this, but we're going to talk about that more on the heart dissection video. Moving on, major landmarks you're responsible to know. So this is a revised list from your lab notebook assignment, but for the most part, everything is still the same. Some things just don't have as much importance as others, and then I did add one extra thing for you guys to know. Don't get overwhelmed. This is what you guys are responsible to know. And then this is what I took off of the list, that it's good to be able to identify them, but you should pay more attention to each of these landmarks of the heart. And so on the next video of the heart dissection, I am going to be pointing out each of these parts of the heart. So honestly, it's not that bad, I promise. If you can understand how blood moves through the heart, you will be able to point out each and every single one of these here. So understand how blood enters and leaves the heart, you'll be able to nail each of these. Okay, so let's go ahead and end this lecture on a homework assignment note. But not just any homework assignment. This is a really cool homework assignment because I had no idea you guys were these really awesome research technicians working in this medical lab. Wow, congrats. That's awesome. And so as you're working with your colleagues, you're processing blood samples, you've noticed that there's this trend that biological males have a greater hematocrit than biological females. Hematocrit, what do I mean by that? So in the medical industry, this is kind of known as the packed cell volume, meaning the percentage of blood cells that are in our body. You've noticed that males have a greater percentage of red blood cells than females. Huh, that's interesting. So what you decide to do is collect some data on that. So you have some random volunteers to give up a little bit of their blood to you so you could collect some data. So what you first need to do is you prick a couple people's fingers, you get a little drop of blood, then you use what's known as a hematocrit tube to suck up that blood. It's kind of like a vacuum effect. You kind of just put that tube up and the blood goes in. Then you place this hematocrit tube in a centrifuge. You spin it down for about a minute, and after centrifuging, you notice that you have this separation here. Well, what is this separation? This down here at the bottom is just the wax that prevents the blood from spewing out everywhere in the centrifuge. This right here are red blood cells. They are condensed red blood cells. You can't really see it, but right here is a very thin layer. It's called the buffy coat. That is made up of white blood cells. And then all of this here, this clear fluid, is your plasma. And your plasma is going to have proteins, platelets, the works. This is what you are going to use to calculate the hematocrit. So in order to calculate that, you measure the proportion of red blood cells over the total volume and multiply that by 100. So here again is just another overview of that. So you have your red blood cells down here at the bottom, your buffy coat, and then your plasma. And I know I said that there was platelets in your plasma, but your platelets are actually going to be in your buffy coat here. So your plasma is made up of water, proteins, nutrients, hormones, etc. So what's important is to measure the red blood cells to the total volume of what is in this tube. And so we are going to be giving that data to you because you've already collected it. You gave it to somebody else to then type up the data, and then we supplied this data to you on your lecture PolyLearn under Lab 4 in the homework file. So you are going to be running this homework in the applet, or this data in the applet, and then completing the homework assignment that will be due on Sunday. Really exciting. And so what you want to know is, what determines hematocrit? If you do actually see a significant difference between males and females, physiologically, why might that be? And so that is something for you guys to investigate and turn in on your assignment due on Sunday. Cool. And that is the end of this presentation. Go ahead and go back to your lecture PolyLearn and let's talk about that heart dissection more in detail. Cheers.